Hello, my name is Murtaza, and today I'll be giving a brief rundown of our poster titled GrapHiC, an integrative graph-based approach for computing missing IC leads. We as a community understand that spatial organization of DNA is important because DNA folds all to itself to form complex three-dimensional structure called chromatin. Spatial interaction and structure are known to mediate cellular processes, particularly gene regulation. For example, a structural reorganization can push an enhancer away from the promoter in a three-dimensional space, where it can lead to gene silencing that can lead to a disease state. To understand the interplay of structure with gene regulations, researchers have developed many techniques. Among them, one of the most commonly employed technique is HiC. IC protocol takes in the chromatin, then waves around the wet lab magic stick and produces something called paired entries. These paired entries essentially contain sequences of the DNA that were found to be co-located or interacting with each other in the 3D space. A common method to represent that information is in form of a contact map of size n by n rows, n rows by n columns, where n represents the number of regions or bins. The entire DNA is divided into in this representation, each observed paired end increments are counted by 1 for the cell that corresponds to the location of the DNA sequences on the paired end read. And from this representation, you can imagine the choice of the size of the bin would be very much dependent on the reads generated to the highest experiment. More the reads an experiment produces, the smaller the bin size we can have, and larger the n versus vice versa. Unfortunately, it's cost prohibitive to conduct a high C experiment that generate a lot of high C heat the cost inter, because the cost um, in terms of both human legend and reagents scales quadratically with the number of reads required for the high C contact map. Because of this, on ENCODE portal, only 92 out of 450 tissue samples have a HiC experiment available. And of those 92 samples, most of the HiC experiment produce HiC contact map with reads in order of millions rather than billions. That is required to understand the structural feature at a fine scale. Although there are other experimental methods that exist and used to study the spatial organization, such as GPC experiment targeting CTCF or finding instruments with particular modification that are known to mediate structure. The data produced through these experiments typically approximate information about the structure. The core insight of our work is that we can utilize these proxy GPC experiments in conjunction with a sparse IC contact map to impute high resolution IC contact maps. To embed both of these modalities in a single representation, we formulate our genomic data as a graph, where each genomic loci forms a node and observed high-C contact provides the edge weight connecting them. Moreover, we embed gypsy signals coupled with positional encodings as our node attributes in the graph representation. We construct the positional encoding vector by picking the top 8 components of the eigenvector of the high-C contact map. These eigenvectors intuitively positions a node that belongs to the same structure together, hereby providing a notion of position to our graph formulation. Next, we pass this graph to our graph encoder, which utilizes a graph transformer convolution to embed our graph in a node-latent embeddings, which we pass off to our graph decoder, which utilizes our unit architecture to compute a dense high-C contact map. We train our deep neural network with mean squared error loss by comparing the output of our model with a dense high resolution high-C contact map and optimize our network to produce realistic biologically informative high-C contact map. To evaluate our model, we compare ourselves against two baselines. First is the high-C-Reg that uses only the chip-seq signals to predict high-C reads, and second, high-CNN that uses a sparse high-C contact map as an input to predict dense high-C contact map. We evaluate our model in 7 different data sets. We use 5 GM12878 experimentally generated high-C data sets with read sparsities ranging from a ninth to a hundredth of the reads, and 2 cross-cell type inputs from IMR90 and K562 cell line. We evaluate our performance across 3 metrics. First is SIM, that compares the visual similarity of the generated contact map with the target high-resolution contact map. Second is Genome Disco, that compares the underlying chromatin topology. And third, we compare the chromatin loops recovered by our imputed contact map with the loops in the target contact map by computing absence score. For all the metrics, a higher score is better, with a score of 1 being the best. On the GM12878 datasets with 5 different levels of sparsity, we find that not only our model GrapHiC is able to show robustness to sparsity of the input data, but it also manages to outperform baselines with improvement on the sparsest data, with the most improvements on the sparsest data set. In a cross-cell type evaluation, we similarly show that our method GrapHiC outperforms existing methods on both IMR90 and K562 cell lines. Lastly, we considered a scenario where we have no HiC contact map available. We show that our model GrapHiC is able to generate a HiC contact map with comparable scores to a scenario where we have a HiC contact map available across all three cell lines that we evaluated it on. Lastly, to test the which part of our formulation helped the most, we conducted an ablation experiment on our graph formulation. And we found that the addition of positional encoding substantially improved our scores in comparison to the Brave baseline graph formulation that does not have any GIP-seq information or the positional encoding. We further found that adding GIP-seq data improved our chromatin recovery scores substantially. In conclusion, our model GrapHiC provides a formulation that combines multiple modalities of genomic datasets into a single representation to impute missing IC reads. We further show that our representation is robust to missingness of IC data, given that we are still able to impute dense IC contact maps. Feel free to reach out on Poster Palooza if you have any questions. Thanks for listening. Goodbye.